Aahana is a professor at the School of Arts and Sciences at Azim Premji University, where she teaches chemistry. Her current research interests involve studying nanoparticle-induced oxidative stress and its role in pulmonary toxicity. Before joining the university, she was a regulatory scientist in the US FDA, Food and Drug Administration. She worked in the National Center for Toxicological Research, developing better regulatory frameworks and methods of evaluating safety and efficacy for cancer nanotherapeutics. Aahana did her PhD in chemistry and material science at the Princeton University, USA. With that, I welcome Aahana.
Thank you. So, good morning, everybody. My talk today is aimed at a slightly higher level, so a high school, college level, right? And actually, I see a lot of my students here in the audience today. Chemistry, physics, biology, right? I've taught them all. And currently teaching a few of them. But essentially today what I'll be talking about is about the intersection of material science and biology, right? I'm not a biologist, so there are very glaring gaps in my knowledge perhaps, right, about biological systems. But I ventured into biology very late in life, right? And I look at it through the lens of a material scientist, right? And these engineered nanoparticles that I'll be talking about today, we are probably being exposed to them in various forms. But today I'll be talking specifically about, in the second half of my talk, about how these amazing properties at the nanoscale can basically be harnessed for cancer drugs, which is my area of specialization. Okay. Now, how I'd like to do this ideally is that whenever you have a question, instead of waiting for the end, maybe you can just put your hand up and ask me, because we have a lot of time and we don't need to rush or anything, and then we can have a kind of more interactive, more discussion based session, right? Okay. So, I mean, this is essentially what a typical nanoparticle, like you can think about a metal particle, right, like say maybe gold, iron, silver, whatever, right, a metal core, or there are other more complicated types of particles, but the simplest one would be a gold particle, right. And then this, obviously this is not really, it doesn't look like a gold particle to you, right, but it's basically covered with what we call ligands, right, a layer of these small molecules or big polymers or whatever that cover it. The moment I inject, and I want this to kind of register, the moment I inject something into your blood, right, your blood is filled with all sorts of proteins, salts, polymers, right. Proteins are essentially big molecules, they're polymers. These are basically going to cover the particle, right, and that basically acts as an avenue for your immune system to clear it, right. So, when we are engineering nanoparticles to go into the human body, right, we find all sorts of ways to kind of disguise or cloak the nanoparticle, and that is by coating it. Coating also, I mean, if I take generally a solution of gold nanoparticles, they'll all kind of come together and clump together, they'll form aggregates, right, they will no longer remain in that nano size. I'll talk about what the nano size is, and so again coating basically keeps them apart, right, keeps them in this kind of colloidal solution form, right. So this is basically what you would consider a typical kind of cartoon picture of any kind of particle inside the body, okay. It's filled with a lot of complicated surface layer, right, okay. So what I'm going to talk about today, initially I'm going to start with what is so special about the nanoscale, right, and when we talk about the effects of materials, how do we think about the effect of nanomaterials differently from the effect of say just any other chemical, right, and then the second half kind of will be a little more technically about taking advantage of the special properties of nanoparticles, and I'll specifically use the example of cancer in this case, right. Okay. So when we talk about nano, right, so how small is nano, right, if you want to get an idea of the scale, nanometer is 10 to the power minus 9 meter, and a human hair is basically about 1,00,000 nanometers thick, 100,000 nanometers thick. So if I look at a tiny gold particle, can anybody, I mean, you can see the size, right? So this is an electron microscope picture. So you have 100 nanometers here, And so when I kind of measure the diameter of this particle, right, it's basically about a 30 nanometer particle, right. So if I take a 30 nanometer particle, say it's a gold particle and it's covered by a typical layer of citrate, which is how we synthesize gold particles, and I blow it up to the size of a football, right, then it's almost basically the same as blowing up the football to the size that is 10 times the moon, right. So that's the scale that we're talking about. That's how tiny nanoparticles are. And this tininess basically comes with its own advantages, because this, apart from other things, the first advantage is that this nanoscale is basically the scale at which biology operates, right. If you look at the size of a typical virus, right, whether it's the HIV virus or the hepatitis B virus, a typical virus is basically in that nano range, right. And if we go smaller, which is the range at which we make, say, quantum dots that won the Nobel Prize last year, right, quantum dots or nanocatalysts, right, like, then we basically go into the domain of the size of, say, a protein molecule like hemoglobin or DNA molecule. That's the size at which we operate, right. Now this comes with its advantages. I mean, it's important to remember that it is the scale at which biology operates, because these particles, in the way they are, without actually dissolving into anything and all, basically can penetrate into your cells. Okay, and that is the advantage it comes with. It sticks to the cell membrane surface and then it can basically be internalized by the cell. That's one advantage. Okay. Now, one example of a nanoparticle that we are all exposed to every day, right, is if you look at, so for instance, this is a typical air pollution monitor, right, which is showing you particulate matter, right. It's showing you, or if you look at an app and you see the air pollutant concentrations, this PM 2.5, PM 10, does anybody know what that is? How do you define it? What is particulate matter? So particulate matter are basically very, very tiny particles of solid materials or sometimes aerosols, which are basically suspended in air, right. So this is an example of a particle that when you get smaller, so this is a human hair, right. So if the yellow particles on the human hair are basically what you would call the coarse particulate matter, right. So I can arrange 5 of them on one strand of human hair, and then on that I can arrange 4 particles of the fine particles, right. And then the smallest type of particulate matter, the classification of the size, is ultrafine particles, which are less than 100 nanometers in diameter, right. So these ultrafine particles are basically everywhere in the air, but we don't measure them. So when we measure air pollutants, we tend to measure the larger particles, the PM 2.5 and the PM 10. We don't tend to measure the ultrafine particles, but they are perhaps the most kind of toxic particulate form of air, right. And even when we are talking about cigarette smoke, right, cigarette smoke also has, like, maybe it's a very, very chemically complicated system. It has over 700 chemicals, right. And when you take cigarette smoke in, you have 2 phases. One is the gases, the gas phase, right, which is easier to study, and then you have the particulate phase or the tar phase, which is very difficult to study. Very chemically complicated system, just like air pollutants, but all basically consists of this particulate matter, right. And the particulate matter, we classify it by size, right, simply because size affects biology. Okay, size affects the biology, size affects the chemistry, and that's basically the bottom line of the entire field of nanotoxicology, right. When we are talking about the properties of these nanoparticles, the size is basically very, very important and very critical. Okay. So again, if I was to compare to the scale of biology, so when we are looking at coarse particles or fine particles, right, look at the sizes. They basically compare to, say, red blood cells in your body, and Staphylococcus is a tiny bacterial cell. So, the fine particles basically compare to Staphylococcus perhaps, right, to bacterial cells. And then when we go to the nano domain again, the sizes compared to the size of these tiny viruses, right. The reason, again, the most basic reason why we kind of compare by size is basically because it affects the amount that a particle penetrates into your respiratory tract, right. Now, the ultrafine particles, which is basically what I study, the ultrafine particles are capable of actually crossing over. They can just slip through the cells of the lungs, right. They first cause a lot of cascade of reactions in the fluid that is there in your lungs, then they can slip through the cells of the lungs, right, slip into your bloodstream and basically get carried everywhere across your body, okay. And that makes them special, of course, because they basically can cross the air-blood barrier, right. Not every particle can cross so easily. They get deposited in your lungs, but these particles are tiny enough to basically go into your blood and then get carried all over your body, okay. And so, something that you are taking in through your lungs can have systemic toxicity in your liver, your kidney, right. And some of these particles, if they are tiny enough, can also cross the most hard to cross barrier that is there in your body, which is the blood-brain barrier. Okay, the blood-brain barrier is something that we have a lot of difficulty in making materials cross, but some of these particles can cross, right. The other thing also is that as you get tinier, the chemistry of the particle, and that is like what I will talk about now, the chemistry of the particle becomes special. Okay. Okay. So, now I want you to indulge me for a minute, and my students have done this exercise in class, but we will do it now together. Have people here seen X-Men, the movie? Yeah? No, yeah, you have. Anybody else not seen X-Men? You've seen? Not seen. Okay. Okay, so a lot of people haven't seen X-Men, but X-Men is a science fiction movie, right? It's a series, it's a comic, whatever, superhero series. And X-Men is basically, so this scene is one of my favorite uses of nanoparticles in cinema, okay? So X-Men is basically a movie about a mutant race, right? And basically these mutants have all these weird special powers. Okay, that has nothing to do with nanoparticles. What it has to do with nanoparticles is that in this scene, this character who is Magneto, right? Magneto is basically someone who can control metals with his power. He has magnetic powers, right? So, any magnetic particle he can control, manipulate, make into any shape and use. And so, Magneto in this film clip is being stored in a plastic prison. Okay, it's a prison completely made out of plastic, and then his colleague or collaborator or whatever, his accomplice, injects metal nanoparticles into a guard's blood. Okay, and then this is what happens. We'll do a small calculation after this. Okay. So what we usually do in class after this is we kind of discuss what the writers got correct. Okay. So what did the writers get correct in this case? You do have iron in your body, right? You have to take iron supplements and all of that for proper biological functioning. So does anybody want to say like what forms of iron you have in your body? Hemoglobin, right? So hemoglobin is right up here, right? I've put hemoglobin up here. So you have hemoglobin, and then any other, do you guys know any of the other forms of iron in your body? Okay, so you have hemoglobin. So the forms of iron in the human body, about 60 to 70%, the majority of the iron is hemoglobin. But then what do you see here? The iron in hemoglobin is basically iron ion that is in a kind of complex, right? So you definitely, magneto doesn't have the power to convert it into a magnetic particle, okay, because it's the wrong form of iron, okay. Then, you know, some amount of iron is stored in the form of ferritin, okay, and then rest is in, you know, other small forms in proteins, enzymes and all, transferrin, but essentially the transferrin is also very similar to hemoglobin. It's a protein and the iron is in ionic form in a complex. In ferritin it is stored, sorry, okay. So it is stored in the form of a mineral in ferritin. So it's a tiny particle of like basically inorganic iron salt, right? And so maybe magneto could use that, okay, to convert, right? So, but then if we kind of see the amount of iron that is stored in the human body, right, that's about 1000 mg in an average human male, which is what the guard is, right. So, if we say that it's 1000 mg of stored iron, can we calculate now whether 1000 mg is enough to make bullets? Do we want to do the calculation? Nobody has pen and paper, but yeah, can you do the calculation? Okay, so anyway, so if we assume that I don't have a board, but if we assume that the diameter of the sphere is basically about 2 centimeters, and so I take the radius of the sphere to be 1 centimeter, then the volume of the sphere would be how much? No? Huh? 4 by 3 pi, right, which is how much in numbers? Roughly 4, right? And then if you say that he makes about, you know, 3 spheres, he makes 3 bullets of that kind, so it's basically about 12 centimeter cube, right? And so if you multiply that with the volume of iron, which is about 8, with the density of iron, then how much iron do you need to make those number of bullets? Sorry? Okay, so 94.5 grams, right? So you need a total amount of 94.5 grams to make those 3 bullets. So he definitely needed the amount of iron that was there in the guard's body in usable form was definitely not enough. So he needed an injection of these nanoparticles, okay? But then the point is that would a person who gets 94 grams of iron injected into the body actually survive it or not, right? Because iron, you need iron, but every element that you need after a certain level becomes toxic, right? So iron also after a certain level becomes toxic, and basically that is what they probably got very wrong, that you could inject 94 grams, 94, right? 94 grams of iron into the human body, but after a certain level, it would show toxic effects. So when we talk about toxicity for an element, when we talk about toxicity for an element, when we say 60 mg per kg, right, we mean 60 mg per kg for body weight, right. So suppose the guard is approximately 60 kgs, right. So the maximum I can give the guard is 3600 milligrams, right, which is 3.6 grams, right. So the maximum I can give the guard is 3.6 grams, but I am injecting how much? 94 grams. So the guard is going to be dead, right? So the guard won't, so that is what the movie definitely got really, really wrong. But there's a catch to it, right? The catch is, can I say that because it's nanoparticles, I'm okay? And it turns out that maybe, right? So the toxicity of a particle of iron in nanoparticle form, and that is the key point here, that the toxicity of a particle of iron in nanoparticle form is not the same as just directly injecting an iron solution into your body. Okay, it's a little more complicated than that. So maybe if I had designed my particle properly, even if it was iron, I could get away with it. Okay. And iron, you know, is basically toxic, and there's a lot of reactions here. But iron and other kinds of metals in the human body are basically toxic because of what we call the reactive oxygen species, right? And reactive oxygen species are just exactly what it sounds like. They are basically molecules, tiny molecules that contain oxygen, that because of the form they are in, either they have like a free electron like this, right? They're radicals, right? Radicals are extremely reactive. So they basically react with everything they encounter in your cell. So iron iron basically gives rise to these free radicals, right? When it is there in a larger than, and it puts the body's oxygen chemistry kind of out of balance, right? That's the reason you age, right? Because you have reactive oxygen species continuously being produced, and at some point of time it goes out of equilibrium and it starts to damage your cells, right? So these reactive oxygen species can be generated even by a particle of iron, but even more so by ions of iron, okay? And that puts the body in a state that we call oxidative stress, okay? So that basically means that so many of these reactive oxygen species are generated, and they are so very reactive, they go and react with everything they encounter in your body, right? They react with the cell membrane, and obviously that damages the cell, right? Because every cell is protected by a membrane. They react with the proteins, and most importantly, they react with DNA, right? So either they kill the cell, and that is what we mean by cytotoxicity. That means they are impairing your cells to such an extent that they are killing the cell. That is the meaning of the word cytotoxic. Or they result in genotoxicity. That means they are not killing the cell, but they are causing mutations in your genes or DNA, right? And what do mutations in your genes or DNA eventually cause? Cell death, yes, but if the cell does not die, then what can happen? Sorry? Diseases, and basically more specifically cancer. Cancer, right, for any cell to become a tumor and to become cancerous, right, the cancer cell, it's basically, it acquires enough mutations that are not repaired by your body that eventually make it a cancer cell. So cytotoxicity is one thing, right, which results in cell death, but genotoxicity is basically what makes something carcinogenic or basically cancer causing, right? That's the meaning of the term carcinogenic. So any ion, so iron ion or even an iron nanoparticle, right? These Fe3O4, right, which is iron oxide nanoparticles, are being explored as a therapeutic agent, right? Because we find that iron nanoparticles can cross that last barrier in your body, the blood-brain barrier, okay? So there is hypothetical research where you can kind of inject it into the blood, it can cross the blood-brain barrier, it can go into the brain and it can dislodge clots, right, under a magnetic field. So it's kind of similar to magneto stuff, right? It's very high tech. You basically put a magnetic helmet on someone, and then, you know, the clots in your brain that are very difficult for doctors to reach surgically, the iron nanoparticle can dislodge, right? That's the theory. So iron nanoparticles are realistic, but any nanoparticle can cause oxidative stress in your cell and then cause all of this damage, and that's how basically nanoparticles basically become toxic, right? Yeah, I can pause if anybody has any questions up to this point. Yeah. Ferritin is the usable form.
So like I would like to know like what makes them usable and what's the difference between them?
Yeah, so I mean it's all my hypothesis, right? The writers of X-Men didn't consult me, right? Yeah, but basically, so in transferrin and hemoglobin, you have iron inside a coordinate complex. Have you studied coordinate complexes? Right? Okay, so I mean it's, you probably studied. So it's basically, if you look at, if I go back to hemoglobin, so if you look at something like hemoglobin, it's basically, this is actually covalently bound to this nitrogen, this nitrogen, this nitrogen, this nitrogen. So it's not in a pure ionic form or in an inorganic form, it's in an organic compound. Okay. So this, the property of it is different from something like iron 304, which is a mineral oxide form. So it will not work as a magnetic particle, right. So what I was saying is that magneto can't use this, right. But magneto, I mean of course it's all hypothesis, but magneto can use something like this because this is basically stored as an inorganic mineral in the form of hydroxides. Okay. So this is an iron hydroxide. So this is an inorganic salt kind of, and that is an organic complex. Okay. It's a coordinate complex. That's all I was saying. Okay. Okay. So now, now we come to the part where we have to distinguish between the toxicity of say just an iron solution. Suppose I take ferric chloride or something and I inject it into your blood versus a solution of iron nanoparticles. Okay. We have to distinguish between the two now. Okay. And for the ferric chloride kind of thing, like I say, I take ferric chloride, ferric sulfate, whatever, I take a salt and inject it into your blood, the toxic dose is very clear. It has been measured time and time again. We know that above this value, iron is toxic. It will kill you. Okay. But for nanoparticles on the other hand, we have to ask some very critical questions. Okay. We can't just say that this is the dose at which it will kill you. Every nanoparticle will be different depending on size and shape, right. So we have to ask, the first thing that we have to understand is that if I am injecting iron or a metal nanoparticle, this is a kind of cartoon picture, it will be a solid core with some layer around it, okay, with some layer of molecules around it. Okay. That is how it will basically look. So I have to ask, the first question I have to ask is, does this particle dissolve in my blood or not? Okay. Does it dissolve in the body or inside my cell? If it dissolves, then my life is very simple. I just treat it the same way as I would treat a solution of iron, because it has dissolved and it is now basically just iron ions or something like that. So that is the first thing, right. Now if it does not dissolve, then I have to ask that fine, there is this solid core, but what else is surrounding it, right. So what other things is it carrying along with it into my cells, because it is going into my cells, right. And the last thing I have to ask, that what effects does the particle itself have? If the particle stays as a particle, does it have any unique biological effects, just like we saw in the case of this, iron particle, which has some unique reactions that it is catalyzing on its surface itself, right? Does this particle also have some unique effects that depend on its size and its shape, right? So, I can no longer just say that the dose is some x mg per kg. I have to ask a number of more complicated questions when I am dealing with nanomaterials. Okay? I can't just leave it at a particular dose. And most of the time this dose has to be different for particles of different sizes and different shapes. Okay? I also have to ask what is the charge on the surface of the particle, because remember that the cell membrane is negatively charged, right? And this is something that you study in 11th and 12th biology or in college biology, that the cell membrane is negatively charged, right? So, for any particle to first enter the cell membrane, it has to basically stick to the, it has to adhere to the cell membrane first, and then only can it be taken in by whatever mechanism. It has to first adhere, right? And if the cell membrane is negatively charged, then the charge on the surface of the particle also matters, right? Because if it is also negatively charged, it will not stick, right? It will just repel. That's simple physics, okay? Right. Now, similar questions actually have to be now asked in real life, right? Because these are examples of products that are actually being sold in the market, right? So, this is a wound dressing. This is actually a product from Vietnam, and this is a product that is actually available in the US, where they are basically putting on bandages, they are putting nanoparticles of silver. Okay. Now, what question do we have to ask here, right? When I am putting silver on my skin, if I just take a silver salt and paint it on my skin, it is toxic, right? Silver is toxic to humans and biological systems in general, okay? But when we are putting nano silver on dressings and then putting them on my skin, the assumption is that it does not go in, okay? Silver does not go in, right? It is staying in this nano form on my skin, right? The advantage of these kinds of dressings, what they claim is that it has very good antibacterial properties. So, if you have like burns that are really, really bad, right, one of the biggest dangers is that the burn will eventually get infected, and this basically avoids that, right. The silver gives it that property, but at the same time we have to make sure that the silver does not enter the body, right, it is not dissolving. So, remember the first question that I asked, does it dissolve inside the body? The first thing that we have to make sure is that a dressing of this kind, the silver does not dissolve in the body, and that is exactly how these things are tested, right. So, you had at one point of time sanitary products like tampons and menstrual pads that were being made by a Chinese company which had silver nanoparticles in it, okay. And then it was studied by the FDA, and it was found that in mice, when you use these tampons, the silver would actually kind of go everywhere in the body, right. So, how you would typically do it is use it on a mouse, then kill the mouse, and then study each organ of the mouse, right, to see how much silver is there in the liver, how much silver is there in the kidney, how much silver actually is there in the brain. And those studies show that these feminine hygiene products, they were actually not safe in terms of the fact that they were putting silver all over the blood, okay. But these wound dressings, now we know that perhaps they are safe, they do not allow silver to go into your blood, so that means we can use them. So, very, very important question to ask is, does it dissolve, okay. Right. Now, the next question that I was asking is, what effects does the particle itself have, right, and does this effect depend on size and shape, right. And now I would kind of like to discuss why the size and the shape becomes important and why the nanoscale is so special in the first place, okay. Right. So, basically when you think about dividing a material up into smaller and smaller pieces, what you are essentially doing, right. So, I am taking a you know, 1 cm by 1 cm cube here, the same amount of material I am dividing up into 1 millimeter cubes. And then when I divide up the same amount of material into 1 nanometer cubes, I am basically increasing the number of the amount of surface area that I am exposing, right. It is basically, you can think about it as simply making a finer and finer powder of a substance, right. The finer powder you have, the more surface area you are exposing. And the surface is important, the surface to volume ratio of a material is important. And the fact that in a nanomaterial you have a very, very large number of surface atoms is important, because the atoms inside a material, right. If you think about, like, say, gold or iron or whatever, the atoms inside the material, they have atoms surrounding them on all sides, right. So if they can make 4 bonds or whatever, they are bonded on all sides, right. They're kind of completely satisfied in terms of their reactivity. But when you have an atom on the surface, right, like somewhere here, it is surrounded by atoms maybe on 3 sides, but not on the 4th side. So it has kind of, like, you can think about it as an unfulfilled reactivity. Okay. So surface atoms inherently are more reactive. So now if I say I'll take this cube, right, and I again divide it up into these 1 centimeter square, and if I show the inside surfaces as in red, when I actually take it apart, you see that I have exposed a lot more of the inside surface, right. And these surface atoms are basically on the surface and they are much, much more reactive. So as I make a, as I make a particle smaller and smaller, right, I am basically increasing the number of particles, the proportion of atoms that are on the surface, and that are these more reactive atoms. So a finely divided nanomaterial, right, is just more reactive than a bulk material. And I think the favorite example of this is gold, right. So gold, we study right from the beginning in school, is a noble metal. Right? Everybody knows gold is a noble metal, just like the noble gases. Gold is very, very unreactive, it's a noble metal. But it turns out that when you divide gold and when you take a particle approximately, say, 3 nanometers in size or 2 nanometers in size, gold actually looks exactly like charcoal. You cannot even say that it's gold. It is black in color, okay, when you divide it up to 2 nanometers, approximately 5 nanometers or less. And it becomes an excellent catalyst. And there is a whole field of study, right, and that's what I studied in my PhD as well. There's a whole field of study of gold catalysis. And gold is not something that we have ever thought of as a catalyst, but it turns out that in the nanoscale, it is very reactive, it's an excellent catalyst. Okay. So that's an example of something that, because it has so many surface atoms exposed, it's such a small particle, the reactivity of it, the way it behaves chemically itself completely changes. Okay. The second thing is, why does the shape matter? Okay. So of course the size matters because there are more atoms on the surface. The shape also matters because you expose different kinds of sites by cutting the crystal differently. So if you look at this shape, the kind of atoms, like there are some hidden atoms here and there are some more exposed atoms here, whereas here, you know, everything is kind of uniformly exposed. So I am basically exposing different types of atoms by cutting the crystal differently. That's all it is. So the shape between this more or less spherical particle and this kind of, you know, polyhedral particle, the two shapes will actually behave differently, chemically speaking and of course biochemically speaking. Okay. So when we are considering toxicity, that's why I am asking the question, what is the size and what is the shape? So now I have to define, is the particle soluble or not? Is the particle, what is on its surface? Okay. What is its size? What is its shape? Only then I can arrive at what is the dose that a human can take. I can't simply just talk about a dose anymore. Okay. That is the key thing. The color of gold nanoparticles also, it's like very famous example, right? it differs depending on the size. So, you can see that these are all, I mean gold you know is yellow, right, I mean that is the gold color, right. But then when you look at particles of different sizes of gold, you see that the colors are basically, you know, from red to purple, and it varies even more. Here when you change the aspect ratio of these nanorods, that means you make them longer, right. So, this is about 2.4, so it is a short nanorod, and this is about 5.7, so it is a long nanorod. When you change the sizes, the color varies. When you are changing the shape of the nanorod, you are making it longer, the color varies from blue to red. Okay. Similarly, when I take a nano shell, that means it has a silver particle inside, and on top of that I have put a film of gold. Okay. And then it basically with the same size, as I increase the thickness of that gold shell, again I can get a brilliant difference in color. Okay. So, this is this color of gold nanoparticles is because electronically it starts behaving differently compared to the bulk gold material, which is just uniformly yellow. Okay. The most famous example of this is of course quantum dots, right, which won the Nobel Prize last year. Again, depending on the size of a quantum dot, you can get all these different kinds of emitted light. So, you are exciting all of them with blue light in this case, but you are getting a range of colors just depending on the size. Okay. So, there are essentially two very different effects. I will just skip over this. Two very different effects, right. So, when I am in a particular size, so here let us talk about everything below 100 nanometers, which is what I am interested in, right. So, everything below 100 nanometers, when I am talking about a very small particle, right, less than 1 nanometers or something, or maybe even between 10 and 1, I am talking about electronic effect. That means the distribution of electrons in the particle is different from what you would see in gold, that is the bulk material. Okay. Now, when I look at a bigger particle, it is basically because I am exposing different facets of the crystal, and I am also I just also have more surface atoms. So, those are what we call kinetic effects, right. And in the middle range, right, between 10 and 1 nanometer, which is the range at which these gold catalysts work, which is the range at which a lot of nanoparticles work, right, you have a combination of the two effects, right. It is electronic plus the kinetic effects. Okay. So, any questions up to this point? Yeah.
Just a thought experiment.
Yeah.
You mentioned that gold in the nano, is it a nano scale?
Yeah. Yeah, it looks like charcoal. It looks like, yeah. No, that's alchemy, not my expertise. No, but no, it looks like charcoal in the sense, I mean like it looks like just this ashy kind of black substance. It's like a black powder, you cannot even tell it's gold. Yeah, that's all I meant. Yeah, sorry. Okay. Yeah. Yeah, so one of them is, the other Vietnamese one isn't. Yeah, so one of them is, yeah. So, these are all tested the same way, they are applied on skin, and then you are basically seeing how much silver is entering the system, right. Okay. Right. Okay. So, now I am going to enter basically the second half, which is now that we kind of know a little bit about the properties that make nanomaterials special, let us talk about how we can use it to our advantage, okay, in biology. Okay. And again specifically using the example of cancer, but again indulge me for a minute, I am going to start with a more material science point of view of how strange biology is. Okay. So, I have a bottle of water, right, and versus a human being. So, suppose I leave a bottle of water on this table and whatever, the room is cold or hot or whatever, what do you expect eventually? Yeah, evaporation, but let us say it is a sealed bottle. What do you expect in terms of the temperature of the bottle of water? it will come to the temperature of the room. That is the simple principle of thermal equilibrium, right, that we know, right, as I mean, it will eventually come to the temperature of the room. If it's cold, it will heat up. If it's warm, it will cool down, right? It will eventually come to the temperature of the room. But what about a human being or any warm-blooded animal or whatever? If I'm sitting in a cold room, 10-degree room, what happens to me? What? Yeah, so we have thermal regulation, right? But at the same time, we have thermal regulation, but then our temperature is maintained at a particular range, right, all the time, right? So there is a difference. So there is thermal equilibrium for a non-biological system, right, a physical system, an abiotic system. It will eventually always reach equilibrium with its surroundings, right? Biological systems, on the other hand, they show this thing of thermoregulation. They are not in equilibrium with their surroundings, but they are basically in steady state, right? So biological systems are at steady state and not at equilibrium, okay? The second thing, if we look at this, so this is just, I'm using the example of temperature, okay, because it's easy to understand. But if we look at it at a microscopic level, right, inside the cell, then essentially any cell is basically a membrane envelope which is compartmentalized and separated from the environment, and inside the cell you have a very, very different concentration of substances, proteins, macromolecules, DNA or whatever from the outside. So that means just because my cell has DNA in it doesn't mean that if I put this cell inside that extracellular fluid that is there in my blood, that also has DNA in it. The DNA is kept within my cell. But if I just took a bag of substance, right, and if I put it, eventually the two things would reach equilibrium, right. So to keep things away from equilibrium, our body is constantly inputting energy and to keep things away from equilibrium and at steady state, right. So it's unequal but constant concentrations of chemicals on both sides of the membrane, okay. Now this thing is what the membrane, right, and this compartmentalization is actually what sets life apart from everything else, okay. So life kind of, if you think about it, begins at compartmentalization, and that membrane formation is actually very, very simple. If you take a solution of these lipids, this is what your membrane consists of. This part loves water, so we call it hydrophilic, right, and this part hates water, so we call it hydrophobic. And just by the tendency of substances to reach the lowest energy possible, right, the part that does not like water, that does not have favorable interactions with water, stays inside, that is the tail, and the part that likes water stays outside, that is the head. And so lipid solution, you take it, you put it in water, it forms this beautiful membrane bilayer, and that is the basic architecture of the cell, right. Every cell is surrounded by a membrane bilayer. Of course, the membrane is a little more complicated, it has proteins and all in it, right, but then at the core it is this phospholipid bilayer. This is the negatively charged layer, right. So when we are thinking of things entering the cell, we have to basically, and if we want a drug or a nanoparticle, whether it is a small chemical or a large chemical like a nanoparticle, when we want it to enter the cell, we have to basically take care of the membrane. We have to have it enter through that membrane, and that is what we are concerned with. We want favorable interactions with the membrane. After that, things progress, and we can think about that at a later stage. But the first stage is get through the membrane, okay? That is one thing, right? So this is a typical question, that is asked in, like, say, first year chemistry or whatever, where you have like a number of molecules and you are basically asked to say what will get through the membrane and what will not. And how we test it in the lab is that if it is to get through the membrane, it has to be soluble in a compound like octanol. If it is to be soluble in your blood, it has to just simply be soluble in water. So every chemist, so a pharmaceutical company will employ a guy called a formulation chemist, whose only job is to test solubilities in both these things, right? That is their only job. They are supposed to make molecules that will dissolve equally in water and equally in octanol or something like that, right? Yeah, a very hydrophobic kind of situation. Okay. Now, that's the first nano drug, right, that was ever made, basically took advantage of the fact that biology compartmentalizes. We learned from biology, we mimicked biology, right, that when you put lipids in water, right, or soap like surfactants in water, they form these nice kind of vesicles. And the first nano drug basically took advantage of that vesicle formation. Okay. And so, I mean, now actually a lot more people are familiar with what we call the liposome, because the vaccines that Pfizer made during the COVID pandemic was basically based on this liposome. It was a liposome with a RNA molecule in the middle, the mRNA vaccines. Okay, you might have read about it. But the first nano drug was also based on the liposome, and it was basically this kind of an architecture. It's called Doxil, okay, and it was approved back in 1995, right. Doxil looks like this on the electron microscope, and you can see the small scale here, 100 nanometers. That means every particle is approximately about 100 nanometers in size or little less, okay. And the Doxil was so special because you see this drug that it entraps, which is called doxorubicin, when it was simply injected into a person as a chemotherapy drug, it was very toxic. It resulted in a lot of cardiac toxicity. That means it basically affected a patient's heart, because it was, I mean, all cancer drugs are basically poisons, right. So, they are chemotherapy drugs, they are all basically poisons. So, it affected a patient's heart, and you can't really give beyond a certain dose, right, without the patient basically having cardiac failure. So, we learned from biology and we protected that drug inside a lipid core, right. So, just like biology compartmentalizes, this drug is also compartmentalized by taking that doxorubicin inside the lipid core, okay. But there is a second layer to it, okay, a second kind of more clever layer to it, which is that when I put things inside the human body, when I inject something inside the human body, a particle becomes covered in your blood by these proteins called opsonins. It's called opsonization. Okay. The particles become covered with these proteins that are all over your blood, and that is what allows your immune system, right. So, you have these cells in your immune system that basically swallow foreign bodies and remove them. They are called macrophages, right. So, it allows macrophages in your immune system to go and recognize the drug or a foreign body, that is what the drug is, and remove it from your body, because it is big enough, it is like a virus, right, 90 nanometers. So, it allows macrophages to swallow it. So, to disguise it is what they call the stealth technology, right, like spycraft, stealth technology. They put a layer of polymer on top, which is shown by this purple layer. Okay. They put a layer of polymer which is based on this molecule called polyethylene glycol, or the PEG. PEG you will come across in every kind of discussion on nanoparticle drugs. It is very, very hydrophilic. That means it loves water, it loves interacting with water, but at the same time layers of PEG are put on all nanoparticle drugs to basically disguise it from your immune system. Okay. So, that is the first liposomal drug that was employed. Okay. How much time do I have? Okay, right. Okay. The second thing that is really difficult about designing biological systems is that designing drugs for biological systems is that they use very few elements, right. So, most biological molecules are basically, we have this entire periodic table available to us as chemists, right. But most biological molecules are basically made out of this carbon, nitrogen, oxygen, hydrogen and phosphorus and sulfur. Okay. So, we use very little of the periodic table, and water is the most common compound, right. This is what we call economy of design in chemistry. That means estradiol, it is the female sex hormone, okay. It is what, and testosterone is the male sex hormone, okay. So, these two hormones have entirely different effects biologically, right. They create entirely different biochemical kind of effects, right. But look at how structurally similar they are. So, not only are you using very few elements, not only are biological systems using very few elements, there is also this economy of design where molecules that are very similar have very different effects, right. And that is a problem of dealing with biology as a chemist or a material scientist, okay. And this is part of a general group of molecules called steroids, right, which includes cholesterol, which is just this basic pattern of 17 carbon atoms arranged in 4 rings, that is all, okay. Another example of this economy of design is morphine, which basically is one of the oldest painkillers known to man, and it binds to something called the opioid receptor. So, you have morphine which agonists, that means it increases painkiller activity, and there is antagonists which are basically very similar molecules, but exactly have the opposite effect, right. That is, they block the effect of morphine, okay. So, you have very similar molecules having very different effects. Now, there is another very dramatic example of this. Here this is exactly the same molecule, but 2 different isomers, that means 2 just mirror images of each other, molecules that are mirror images of each other, while the R enantiomer was basically a therapeutic agent, the S enantiomer is a teratogen, that means it causes birth defects. So, the thalidomide tragedy of the 50s is basically emblematic of this fact. So, in the 50s, thalidomide was prescribed by doctors all across Europe and in the UK to expectant mothers to treat morning sickness, right. But what they did is they prescribed a mixture of these 2 drugs. They did not isolate just this one, because they did not know about it. So, they prescribed a mixture of these 2 drugs, and it resulted in hundreds of babies being born with very serious birth defects or dying or whatever, because it was a teratogen. A teratogen is a substance that causes birth defects or mutations in an unborn fetus, okay. And this is actually where the US was kind of leading the whole drug regulation, because there was one woman in the FDA in the US who said that no, we do not have enough evidence, and therefore did not approve this drug, right. So, but the main point is that very similar molecules that are exactly identical, but just mirror images of each other, as you can see, can have such very different biological effects, where one is actually a helpful drug and the other one is very devastating in terms of its effects. So, this is what we call design economy, right. Now, this design economy becomes even more prominent when you are dealing with cancer drugs, right, because you can think about 2 types of drugs, right. A drug that kills a bacteria or a virus, and we do not have virus, but okay, a drug that kills a bacteria or a fungus or some other organism, or a drug that produces a physiological response, right, that basically is a painkiller or an anti-allergy medication or whatever, right. Now, These drugs are easy to design, right, because it's killing a foreign organism, it's creating a bacteria. But a bacteria has a very, very different proteins and all the other molecules that are responsible for its function compared to you, right. So a drug for a bacteria is very easy to design, okay. Whereas anything that produces a physiological response is slightly more complicated. But a cancer drug is very, very complicated because a cancer cell is basically your cell, okay. It is a cell, a human cell that has gone rogue. That means because of mutations in its DNA, it started basically dividing uncontrollably, and then it has basically become a tumor, right. It's not stopping growing, and that is what a tumor is. So a cancer cell is obviously very, very difficult to design a drug for, because you have it is extremely similar to your healthy cells. So when one is designing a cancer cell, a cancer drug, the obstacle is that it is very similar to a healthy cell. So what you are trying to do is basically prevent toxicity. That means what we are trying to do is prevent it from killing. So all the kind of really, really devastating effects that you see of radiation therapy or chemotherapy is basically because it's killing the healthy cells in your body as well as the rogue cells, because it has no way of distinguishing. So nanotherapies are basically a way of trying to distinguish between the two, okay. Then the next part is basically this rapid evolution. Because it undergoes so many mutations, tumors can rapidly evolve, right. So what is a correct drug at today may not be the correct drug 1 month from now. So that is also something that you have to contend with, okay. So really, I'm not going to go into more detail, but when we talk about the requirements, we are trying to basically target a cancer cell, right. So we want targeted drug delivery, we do not want it to basically affect healthy cells as well. So what the 4 kinds of things that is cancer nanotherapies are used for is, one is targeted drug delivery, the other one is radiotherapy enhancement. That means that when you are given radiation therapy, you are basically exposed to very, very high doses of radiation, which kills everything, because radiation does not discriminate, right. But the radiation therapy enhancement is basically putting these gold nanoparticles into your cancer cells selectively, where they act as an antenna for the X-rays, right. You can think of it as an antenna. It enhances, it is a sensitizer, it enhances the effect of the X-ray. We will look at an example of that. And then we look at an example of something that is very new age, that is the first gold nano drug that has been approved by the FDA, which is used for photothermal therapy, okay. And I will come to what is photothermal therapy a little bit later. So in terms of the conventional approach, right, you are given just higher and higher doses, you are given harmful radiation, you are given combined drugs where the dosing becomes complicated, and you are diagnosed later and later. But with nanotechnology, now what we are thinking of is targeted delivery, right. The radiation antenna kind of enhancement, sensitizer inside the tumor itself, and then you have drug particles which carry multiple drugs at once. So the dosing is easier, and then early diagnosis, which I am not going to talk about. So there are lots of different types of drugs that are being explored, lots of different types of materials, lots of different types of shapes, lots of different types of surface chemistry, lots of different types of sizes. Now these are the only 2 types that are actually currently market approved, okay. Liposome, and now recently a gold nanoparticle has also become market approved. I will talk about that, okay. So liposome is the example of doxil that I gave you earlier, okay. So, two promising types of materials, one is a liposome and the other one is a gold nanoparticle, which I can basically decorate with all sorts of things on its surface. Okay. So, there are two types of targeting that we can talk about. One is passive targeting and the other one is active targeting. Okay. And Doxil is an example that I used earlier, and that is, this is an example of passive targeting, right. What does passive targeting mean? Passive targeting basically means that because your tumor is a very highly kind of evolving and growing system, and it is growing at a very, very rapid rate, the number of blood vessels in your tumor is just much higher than any other part in your body. Okay. So, by passive targeting, I let the blood vessels of your body do the work. So, that means by just simply keeping that nanoparticle in circulation long enough, I eventually let it reach the tumor site, and it basically stays for longer in the tumor, right. And then it generally, just by law of numbers, statistically there are more drug particles at the tumor than anywhere else. So, it is completely just using the fact that you just have more blood vessels in the tumor. Okay. So, that is passive targeting, and for this is basically a drug that is used for passive targeting, and it is called enhanced permeability and retention. It basically just circulates in the body long enough, reaches the tumor site and stays in the tumor long enough. That is all passive targeting is. Active targeting is not something that has been successfully clinically approved, but the principle is that there are certain markers on every cancer cell that are overexposed, overexpressed, right, because of the biology of the cancer cell itself. So, we identify those proteins or markers, and then we modify the surface of a nanoparticle with something that selectively binds to that protein. And so, this gets targetedly internalized into the cancer cell, and this is basically what active targeting is. This is something that is being explored. There are lots of things that are in the animal phase, but nothing has actually materialized to the clinical phase yet. Okay. So, you can think about decorating gold nanoparticles with all sorts of different compounds. You can think of doing photothermal therapy with the gold particle itself. Photothermal therapy is basically exposing gold particle to light, and then the gold particle produces heat inside the cell. I will talk about that. And then radiation therapy, whereas as I said, gold particle itself acts as a kind of antenna for X-rays and results in a cascade of chemical reactions in the cell, which basically selectively kills the cancer cell instead of everything else. Okay. So, this is an example of animal study where you can see this is decorated with a targeting ligand. Okay. So, it is decorated with the targeting ligand, and you can see that in a mouse there is accumulation. This is a non-targeted gold nanoparticle. This is probably going to accumulate a little bit because of that passive targeting that I talked about, and when you have active targeting, you see that it has accumulated quite a bit more. Okay. So, this is one example. Then the first gold drug that I was talking about, that basically is called Orolase, right. It has been approved. It's basically a group in Rice University in the US that have developed it, and it's a gold shell, and when you put it in the tumor and you basically put near infrared radiation in the tumor from a laser, this particle produces heat. So, you can see how, I mean, ignore all of this, but you can see the amount of heat that is being produced around the particle is being measured here. So, as a result, what happens is a tumor irradiated with something that is actually quite harmless, like near infrared light, this is in the mouse, completely disappears because of this targeting inside the mouse itself, right. So, I mean this is the data, right. So, you can see the green is basically the targeted oralase therapy. This is the tumor dimension, that is how we measure whether a drug is effective or not in cancer. This is the tumor dimension, and then this is it's 0 basically at day 10. And here also you can see the survival rate of the animal controls, right. So, the control group is basically without any treatment. The sham treatment is where you are just putting the laser and nothing else, okay. And then this is the treatment group where you basically can see 100% survival after even day 56 and all, right. So, all of the animals basically survive. So, that is oralase which has been kind of. So, there are some unresolved questions, right. The targeting has still not been achieved, right, and the failure rates for nano drugs are very, very high, okay. So, the main point is that there are lots of different physical properties that need to be known. Whenever you produce an engineered particle, it will always be little bit variable compared to like a small molecule drug. And nanoscale, the very special property of nanoscale means that even a small change can have a very, very big biological response. And so, that is why I think the failure rates of these drugs are very, very high. So, to end, I was thinking that where can the next generation of students basically contribute, right. So, of course, from the biology side, we need to understand tumor angiogenesis or the growth and the development of a tumor better. We need to identify these new receptors, biomarkers which we can use for targeting. In chemistry and physics, of course, you need to get better methods of characterization. You need to design new nanoparticle architecture. And then the last kind of overlooked thing I think is that we need to do away with having a lot of animal models, which basically make the development of a drug very, very expensive and also a little bit inhuman, right. So, if we can come up with like sort of models using machine learning, which is a kind of emphasis that many scientists are kind of pursuing now, where instead of exposing an animal model, can we actually make a computer generated model to model the same kind of exposure, right. And this would especially be useful in the case of tumor treatments, because essentially how you treat any of these tumors inside an animal is you use a very severely immune compromised mouse, okay, and you inject it with human cells, right, and then you basically explore how the tumor develops, because you are basically testing a human tumor inside an animal, right. And so, there are many ways of making these immune compromised mouse or giving the mouse an human system, human immune system, which are not really humane. And especially in the first phase of animal trials, we do not even need to follow what is called good laboratory practice guidelines. That means we do not need to legally take care of the welfare of the animals so much, right. We can do a lot of what we want, and that is at that point of time pretty inhumane. Yeah. So, this would also be a kind of area to actively explore, right. Can we make predictive toxicology models? So, I will end here and yeah, okay, thank you.